Dr. Pete Smith. Hi, Pete.
Hello, everybody. Hello, Jane. Thank you.
Hello. Dr. Daniel Tillett, who's Race's CEO and Managing Director. Hi, Daniel.
Hi, Jane. Hi, everyone.
Hello. And tonight we also have a special cameo from Race's Vice President of Research, Professor Mike Kelso. Evening to you, Mike.
Hi, Jane. Hi, everyone.
Hi. Okay, so housekeeping. The format for tonight is that Pete, Daniel, and Mike will run us through a presentation. It's been lodged just now with the ASX. A word of warning, it will contain some science, but it will also contain the so what from a commercial perspective on Race's announcement regarding significant IP progress this week. So after that, after we've made it through the presentation, we'll open up the floor to Q&A. If you'd like to ask a question, you can do so using the Q&A function in the ribbon at the bottom of your screen. We'll get through as many as we can in the time that we have, and we've earmarked about an hour for tonight's webinar. So with that, I will ask Pete for you to start your screen share and then pass across to Daniel to get us on the road. Thanks.
Thanks, Jane. Next slide, please, Pete. So I'll give you a brief overview and then pass it on to Mike initially and then on to Pete as well, and I'll come back for a bit more later on. We really want to just cover a few key points up front, make sure you sort of take away this. There will be a lot of science. It may get confusing at times. Don't worry about it. If you don't understand, it doesn't matter. What really you need to focus on as an investor is the commercial potential and what this all means. So the simple science side of things is that bisantrene was found to consist of 3 photoisomers. This has been known by us for a while, but it wasn't known by anyone else. This has enabled us to work out how to keep it in the form that's most active and also how to go about patenting this discovery. So we've filed 3 patent applications covering the chemical structure of the isomers, how to make those isomers, how to formulate them, and how to use them most effectively. If these patents are granted, which all the advice we've received to date is that they are likely to be granted, but of course we all need to know as we work through, we'll have 20 years composition of matter. This has significant implications not only for our current RC220 formulation, which is in phase 1 trial at the moment, but also for the original formulation RC110, and Pete will talk more about that later on. Ultimately, it comes back to what biotech is all about is identifying new IP, developing that IP, proving that out, and generating real value for shareholders. And this is a major step in that direction. And on that, I'll pass you over to Mike to give you a lesson on chemistry. Thank you.
Thanks a lot, Daniel. So I'm going to start off my presentation just with a very quick introduction to some chemistry. So shown on the left-hand side of the screen there is the structure of bisantrene, written in a longhand notation where we show all of the carbon atoms, all the hydrogen atoms, and all of the nitrogen atoms, and all of the bonds that link those atoms together into a scaffold. And you can see some of those bonds are single lines and some are double. They're single and double bonds. So what that structure represents is the molecule of bisantrene. And in fact, composition of matter pattern is what is shown on the screen. That's what gets covered, is that particular architecture of how all those atoms are linked together and how that determines how they function and why bisantrene does what it does. On the right-hand side is the chemist representation in the form you would have seen on the ASX release. Much simplified, all those hydrogen atoms are missing, but as chemists we know they're all there because of the properties of the atoms involved. So next slide, thanks. Okay, so what we're showing here on the left and highlighted in orange there is what we call EE bisantrene. So that's the structure that you always see drawn for bisantrene. I'll draw your attention to the red parts that show E next to it. So they're the important bonds for this discussion. Those carbon-nitrogen double bonds are called hydrazone bonds, and theoretically they can exist in in two forms, both E or Z. And historically, bisantrene has always been assumed to exist in that EE form shown on the left. But what can happen is, or what can potentially happen is, I've got a molecular model here, and you can see we've got the 3 rings of bisantrene shown down the bottom. I've left off the bottom half just so I can hold this model. But the important part is this bond shown here where my finger is touching. That's the carbon-nitrogen double bond. It's shown currently in the E configuration. If we move it to the Z, all that means is we're pulling this part of the molecule off and we're reattaching it down here. And now we have a completely different molecule because this group now is very much interacting with this structure down here. So when I say theoretically that can happen, meaning that these bonds can exist in E or Z forms, but in reality, most times hydrozones are almost always in this E form. So what we're showing here is the theoretical possibilities for bisantrene isomers. So next slide. All right, so just going to give you a bit of the history around the discovery here. So it all dates back to an early toxicology study the company was doing, which is where you administer the drug to animals and you study the effects of the drug. Part of the study involves taking blood samples from the animals and analysing those samples using liquid chromatography mass spectrometry, or LC-MS. And what you do that for is so that you can understand what the concentration of bisantrene is in the blood at the time at which you take it. So when we did this experiment, what we noted was, firstly, some of the samples look just like we'd expect, where a single peak comes out, shown on this chromatogram, LC-MS, and that symbolises that's EE bisantrene. But what we also noted was that in many of the samples, this second peak was appearing, and it was appearing in really unpredictable amounts. Sometimes there was quite a lot, like the middle one, sometimes on the right-hand side a little bit, and sometimes none at all, like on the left-hand side. So one of the other things that we knew about this peak that was appearing is it had the same mass or molecular weight as bisantrene. And that immediately tells us, well, it may well be an isomer of bisantrene. What that means is it's got the same number of carbons, hydrogens, and nitrogens in the molecule. And so initially we thought, well, perhaps this is forming in the animals themselves. But what we then thought is, well, that's probably not happening because it was so variable, the amount of this new peak that was forming, you couldn't explain it using the principles of metabolism. So that led us to think, well, perhaps there was an explanation here that involved exposure to light, where the samples that were going into the animals, so the infusion solutions, were receiving variable amounts of light exposure prior to going into the animals, or potentially the samples when they were coming out of the animals in the blood, those blood samples were being exposed to light to variable amounts before they got analysed. So that was the thinking at the time. And then so we had to go off and do an experiment to try to figure out if that was the case. And we did this one here. So what this experiment involves is you take a sample of EE bisantrene in water. So you just dissolve it up and it's pure EE bisantrene, what you start with, and then expose it to light for varying amounts of time and monitor that solution by an analytical technique called HPLC. And so when the sample was protected from light, so no light exposure, we see the expected chromatogram, only a single peak, meaning that's EE bisantrene. But after 15 minutes of light exposure, so that's a very short time, and this is lab light exposure, so ambient light, we can see the appearance of this new second peak. And then if we have half an hour of light exposure, you can see that now that second peak is as big as the first one, meaning you've effectively got a 50-50 mixture of those two compounds. So this experiment made us pretty convinced that it was ambient light exposure of bisantrene, EE bisantrene in solution was forming this second peak. So at this point, we didn't know what it was. We had some suspicion that it was an isomer based on the molecular weight. But what you've got to do then is you've got to isolate a pure sample of the new peak that was coming out. And so we did some work and we figured out a way to get a pure sample. So the chromatogram shown on the left means that's one peak, that's one compound, and that's our unknown second peak. But lo and behold, when we took that sample, even stored in the dark, it re-equilibrated or reformed EE bisantrene really quickly. And you can see there's two peaks there showing, indicating that was happening. So eventually we figured out that we could solve that problem for the purposes of the analysis just by getting the analysis immediately after we purified that peak. So we couldn't wait at all. And then so we took that sample to the machine, which is called a nuclear magnetic resonance spectrometer. And we created this thing as shown on the screen called a spectrum. And what this spectrum shows us, all these peaks that you can see, they correspond to the exact molecular architecture corresponding to EZ bisantrene. So it was absolutely unambiguous proof that that second peak that was appearing was EZ bisantrene. Right, so just to summarize what we're talking about then, we start off with E bisantrene. Both of these carbon-nitrogen double bonds are in that E form. Visible light is shone onto the solution of those molecules, and then we get this conversion over to EZ bisantrene, the first one shown in the middle. Okay, and those arrows that you can see show interconversion. That means that, once it gets to EZ, it can actually go back to EE and it eventually reaches what we call an equilibrium. You can also see that on the right-hand side, there's a ZZ isomer, and we do see this sometimes, very small amounts usually. It's not really part of, the major part of the game that what's going on here. It's really about EE and EZ bisantrene. Okay, so now that we knew what the structure of our mysterious new molecule was, the EZ bisantrene isomer, we wanted to know how active it is as an anti-cancer compound. And so the way you would normally do that is you would isolate a pure compound, EZ bisantrene, and test its ability to kill cancer cells. And I've already shown you, though, that we couldn't get hold of pure EZ bisantrene easily because it re-equilibrated back to EE. So what we thought we could do then is, okay, let's just create varying mixtures of the two isomers and test their anti-cancer activity. That means their ability to kill cancer cells. And we chose this breast cancer cell line MDA MB231 to do this experiment. So shown underneath there, you can see 0 hours. That means there's been no light exposure on a sample of EE bisantrene, 100% EE. If we did the 15-minute exposure, 0.25 hours, converts it to a 73 to 27 mixture, and so on. You can see increasing amounts of light cause increased amounts of EZ bisantrene in the solution. So we take those solutions and we treat these MDA MB231 breast cancer cells with the compounds, and we see what they do relative to one another. So shown at the top right-hand side there on the graph, we're plotting cell viability. So that's effectively, think about that as the number of cancer cells that are alive in a solution. And on the bottom axis, the x-axis, we're showing increase in concentrations of drug or mixtures in this case added to those solutions. And following the black line, you can see when the concentration is low, all of the cells are alive. But as you increase the concentration from left to right, you see the curve goes down, and you reach a point where 50% of the cells are still alive, and that's the point we call the IC50. And so that tells us how much drug is in solution to cause 50% of the cells to be killed. And of course, you want that number to be really, really low, because that tells you that your drug, you don't need a lot of it to kill cancer cells really well. So when there's no EZ bisantrene present in the mixture, we see the number there is 265. When we have a bit more EZ, that number goes up to 310. And then when we're starting to get really significant amounts of EZ isomer, 482, and then finally 824 nanomolar. That is the unit of that measurement. So that tells you how many molecules are present. Just down the bottom, this is a plot showing those values again, IC50 for those 4 different treatments versus the amount of EZ-bisantrene present in the solution. And you can see, if you follow that line from left to right, that the number goes up and up and up, the IC50, telling you that as you have more EZ-bisantrene present, your ability to kill those cancer cells is diminishing greatly. And while we couldn't determine whether there was any activity of EZ-bisantrene because of its properties of re-equilibrating back to EE, this graph shows us, you can see by the rate at which it's accelerating northwards, that if you got to 100% EZ, you're ultimately going to be finding that that compound has got very little, possibly no, anti-cancer activity. Okay, so that's told us that EZ-bisantrene against that one particular breast cancer line is poorly active or low activity, but that's only one cell line. So we thought, well, let's try and see what happens across a really broad range of cancer cell lines. So we've tested 143 cancer cell lines representing 23 different tissue types. So that's covering things like pancreatic cancer, stomach cancer, lung cancer, skin cancer, all sorts of different cancers. And so we do effectively the same experiment that you just saw, and we measure the IC50 values, and we measure it on a pure EE-bisantrene solution. So you can see 100% EE at the bottom. And another one we've purposely isomerized to 58 EE, 42 EZ. And then we treat each of those 143 cell lines with both of those solutions separately, and we measure the IC50. And on the graph shown on the right, if you concentrate on the EE-bisantrene column there, each of those dots represents one of those 143 cell lines, and it's IC50, if you read it off to the left there on the y-axis. And the same for the mixture. And what's really telling here is the line shown in both of those columns, which represents the average IC50 across that whole cell panel. And so on the left for EE-bisantrene, we see that it is lower than on the right. And that tells us again that on average, EZ-bisantrene containing mixtures are less able to kill these cancer cells across the whole panel. Okay, so that's some of the experiments we've done, and just to give you a flavour for how we made this discovery and what we know about it. So I'm going to pass it over to Daniel now, and he's going to tell us about the patents and the value that they bring to race.
Thanks, Mike. So this brings us back to this composition of matter IP again, and I'll take a little bit of time to explain why this has so much value to the company. So composition of matter claims protect the chemical structure. So we've been through, from Mike, quite an extensive description about the actual different chemical structures that bisantrene can form. So we now can have claimed we've made this discovery, it wasn't known before, and we can actually claim that only the EE bisantrene form is active. These are the composition of matter claims in patents, are the strongest possible claims you can get in the pharmaceutical industry. They basically protect your drug molecule from any infringement by any generic manufacturer or other manufacturers. So people basically can't go out and sell your drug without your permission or licensing it from you. As a consequence of that, this drives a lot of value for a new drug with composition of matter IP around it. Means that anyone who acquires the patent or purchases or licenses the patent from you doesn't have to worry about someone else competing against you. And if you think about the difference between new drugs and how expensive they are, oncology drugs can be easily over $100,000 per patient per year versus a generic drug which might be $50. You can see there's a huge difference in the value of having a composition of matter. And the only thing that protects that $100,000 pricing is composition of matter IP. So it prevents generic competition. There's also the potential there of having an additional patent extension that it pretty much only applies to composition of matter IP. That gives you, instead of the 20 years, you can have actually 25 years. So that's potentially very valuable as well. And you can't work around composition of matter IPs by changing the formulation. So you can't just make a new way of packaging up the drug and selling that. This means that you're really stuck. You have to wait till the end of the composition of matter IP. And generic manufacturers always wait till the end of the composition of matter before they introduce a new drug onto the market. You can think about all of this is that the patent life becomes critical to the value of any IP, but it's particularly relevant to drug IP because it takes so long to bring a drug to market. So between the original discovery of a new drug, the original chemical discovery in the lab, and by the time it's sold to the first patient, could be 10, 12, 15 years. All that time as a pharmaceutical company, you're spending money developing that. You're making a loss continuously, and then you only get to recoup on that investment after a long period of time. And so the longer that you have to recoup that, the more value there is to the drug. And Chris is basically what's called the net present value of the drug. Pete, next slide. So people have asked, and I know there's probably a few questions in the seminar, is, how likely is it that we're going to have composition of matter patent? And ultimately, the only way to find that out is to submit the patent and wait for the ruling. But what's required for a patent? So you can get a pretty good idea whether a patent's likely to get through, and we can get advice from people skilled in the art of patent writing whether your patent's likely to get up. You need three things. It needs to be novel. It needs to have utility, it needs to be useful for some purpose, and most importantly, it needs to be non-obvious. And it's not non-obvious to an average person, non-obvious to someone who's skilled in the art of the invention, and that can be quite difficult. So you need to have, if you have a new chemical matter, it needs to be not obvious to anyone who's a skilled chemist. And the discoveries we've made around bisantrene fit all three of those. So the novelty, no one knew about the photoisomerization of bisantrene. They certainly didn't know that only the EE form was active, so you got some utility there. And the non-obviousness, so most chemists, as Mike mentioned, would look at the structure and would not think that photoisomerization and stable photoisomerization would be something that would occur. And it's only through careful follow-up of research and really putting effort in that we've been able to get to that point. And that really does move it a long way from doing something that you would see straight away if you were a skilled chemist. We've filed 3 patents around this discovery, one on the structure and the isometric forms, isomer forms of bisantrene, so all the discovery of the photoisomers. That's the composition of matter patent. We also have ones around administering the patent and keeping it in the form, the right form. And we also have around the process of manufacturing pure EE bisantrene at scale, which is not an easy thing to do. There's a lot of complex chemistry and other processes involved in actually ensuring that you get pure EE bisantrene, and you don't want to be injecting some sort of crude mixture into a patient. You want to be injecting a very high purity drug, and that's not a straightforward process. So this brings us to these mixtures of isometric mixtures. Why can't you just go ahead and use the mixture? You could just make up, add a bit more because some of it's converted to EC, and you just increase the dose to the patient. Apart from the fact that, of course, because it's due to exposure to light, you'd have a problem because some patients would get a small amount and others would get a large amount. But really it comes down to the regulatory agencies, the EMA, the FDA, TGA do not like mixtures of different isomers. These are called isometric mixtures, and these drugs have a real problem. They're very common, particularly for older drugs. But the problem with them is they have a whole series of sort of bad effects that you don't want in a new drug. Ultimately, if you put a mixture of something in, you're going to have to, and one of the forms is active and the other one's inactive, you have to put more drug in to make up for it. So you have to, increase the dose, which can be a problem. But the real major problem is that the inactive forms or the other forms that you don't want, they aren't free of toxicity and side effects. And so they can have real problems for a patient. It may be that the active form is totally fine for a patient and they have a very bad reaction to the inactive form. So if you put a mixture in, they'll get that side effect that you don't want. It also causes a lot of problems with drug-drug interactions. So many patients are on multiple drugs at the same time. Every time you add a new drug to other drugs, you increase the risk of drug-drug interactions and side effects, and they can be very serious all the way through to death. And finally, it's very difficult to study these drugs in patients if they're made of a mixture, because those mixtures behave differently from one another. So as a consequence of all of this, the agencies really pretty much demand that you have a mixture of drugs. Not a mixture of drugs, have only a pure isomer in your formulation. As a consequence of that, there is a whole series of IP implications around that, and there has been some examples of where moving from mixtures of drugs to pure drug has actually extended the life of a patent or created a totally new patent. Plavix, which is a anti-clotting drug, was originally patented back in 1983, a long time ago, as a mixture of the two forms, the S and the R form of the isomer. And then it was only patented later, 5 years later, as a pure isomer. It was discovered that that was the active form and the R form didn't have any activity. As a consequence of that, when they took the pharmaceutical company that owned it, took it to the FDA, they got it approved as a pure isomer, and of course the FDA was happy about that. And ultimately, over the lifetime of the patent, it generated, almost 43 or $43 billion, and this is in, money before 2000 when a dollar actually meant something. As a consequence of that, that extra 5 years added an enormous number of billions of dollars onto the value of that drug. Lexapro is another interesting example. This is a drug that's used in treatment of depression. It was proved as a mixture originally. It wasn't known. And then later on, it was discovered that only the S form isomer was really had any activity, 100 times more potent than the R form. And then that discovery was patented. And that went on to be the S form was approved as a pure S form. And over their patent life, almost $14 billion in revenue. So you can see here that doing these types of activities can generate very, very, very large returns for investors. And it's sort of the sort of thing that you would like to discover. And at that, I will pass it over to Pete to talk a little bit more about the commercial significance and his experience over the years with this exact topic.
Yeah, thanks, Daniel. And I just want to take an opportunity to congratulate Mike and the team down at the University of Wollongong for the incredible work they've done and the amazing amount of value they've added to our business. So looking at this patent, these discoveries from a commercial point of view, this is basically commercial gold. The ability to patent this and get composition of matter claims on a 45-year-old drug is really quite remarkable, first of all. And it really then shuts out any arguments that somebody might possibly develop a competing formulation to RC220. And whilst we've always maintained that we thought that was very unlikely, and I believe that genuinely to be the case, Big Pharma will use any big stick they can get their hands on to try and beat down the price in any negotiation. So this makes our position rock solid. There is no way that anybody will be able to come in and follow our path because of this patent. So it's very, very important for RC220, and obviously it adds to the building clinical data we've got. We've already established, only N of 2 patients, but we've already established that it is safe to administer through a peripheral vein, and so far so good in terms of the safety in the study. So the data is building, and in a very positive way. Now, on to maybe a more significant change, and that is RC110. Now, this drug has worked in AML, where the original bisantrene was approved in France in 1988. We have seen in a couple of studies a very, very good response rate in recent studies conducted in Israel that RC110, bisantrene, can get a 40% overall response in AML as a monotherapy, but in combination in much later stage patients, it also showed a 40% response rate. And that's very, very similar to the data, in fact, identical to the data Lederle generated. And just looking overall at the activity, so there's a whole range of drugs here with different mechanisms, IDH1 inhibitors, IDH2, menin inhibitor, and there's a chemotherapy drug, cytarabine. And what you can look at here is the response rate, and bisantrene, RC110, compares very favorably to most of them. But very importantly, it can be used across all of the patients, whereas some of these drugs are really confined to quite small patient populations who have a particular mutation that is being targeted. So we think that it has a role to play. Cytarabine, and again, a broad chemotherapeutic agent, again, similar, but doesn't have anything like the overall response rate. And the point here is that we have phase 2 data with RC110, and so it theoretically could enter, once all of the paperwork's been filed, it could enter into a registrational phase 3 trial. And we're just really starting to look at the ramifications of that. Now, in terms of partnering, there's some complexities here, because if we've got RC110 being delivered by a central line and then RC220 becomes available, there is a risk of cannibalization. But we think the opportunity here can be to go to a potential partner with something that's phase 3 ready. And, again, just to repeat, it is really amazing to have a phase 3 ready compound with 20 years of patent life. I mean, that just does not happen. So this could be quite an interesting, exciting opportunity for someone to take RC110, but then also to be partnering on RC220 so that they can get something onto the market very quickly in AML and then follow up with RC220 in solid tumors, and then maybe transition to 220 in AML as well. So I'm just really starting to think about this, particularly now we're getting the safety data coming through on RC220, and we've got all the safety data, obviously, from RC110, which was very well behaved. So in conclusion, and then we'll be opening up for questions. Yeah, we've shown these 3 isomeric forms, EE, EZ, and ZZ, and just again to reiterate, this photoisomerization was not known. And again, getting back to the strength of the patent, the fact that 45 years has passed and nobody's discovered it, and then Race has discovered it, actually adds a lot of strength to what we're doing. It looks very much to us that only bisantrene has any anti-cancer activity. I think looking at the data, the other isomers do not. And we have used bisantrene in our formulations with 110 and 220. So all the data in the modern era was with the bisantrene. And we go to a lot of lengths in the RAC010 protocol, the current study, to protect the drug from light before it goes into the patient, and then to protect the samples. So we don't have a situation like Mike showed earlier on, where we're losing some of the bisantrene because it's being converted to another isomer. So we filed the patents. We've been told by counsel that they feel we've got a very good chance of getting claims up, and we're actually going to be going for what's called an expedited review. So this will we can very rapidly, and it happens in about a period of about 8 weeks, we can actually get an early assessment of the likelihood of patentability. And there are always questions in those. They never come back clean, but it will give us a pretty good idea of what sort of counter arguments we'll be up against. So with that, I will hand back to Jane, and we can get into some questions.
Thanks. Excuse me. Thanks, Pete, Daniel, Mike, for taking us through the science and also the commercial potential behind the work that's been done here. I'd like to add my congratulations to the team for getting to this point. It's big news. So with that, I'd now like to open up the floor to Q&A. If you'd like to ask a question, the procedure is to pop your question in using the Q&A function in the ribbon at the bottom of the screen, rather than using chat or raising your hand. Thanks to the many who have submitted questions already. So let's start with this one, perhaps for you, Mike. Although the EE isomer is novel, if the processes to create and store it aren't novel, why would those claims be allowed by the patent examiner?
Okay, thanks, Jane. That's a really good question. In terms of creating bisantrene, in the past, people have synthesized bisantrene. That's not new, certainly. And what you buy from a chemical vendor in a powdered form will be bisantrene dihydrochloride, the salt form. But what we've found is that when we tried to scale up, because we're talking about synthesizing multi-kilogram scale bisantrene dihydrochloride, using the known methods, which is performing the last reaction in a solvent called isopropanol, that reaction wouldn't go to completion. There were byproducts formed, really created a lot of problems. So we had to find innovative solutions to performing that final step. And so we switched to doing it in a different solvent, methanol, and we found some interesting things like there was more isomer formed when we made that switch. However, the reaction worked better, less side products, and we figured out a way to push that EZ isomer form back to all bisantrene in the process. So all of that means that there's innovative discoveries were made and allowed us to file one of those 3 patents is in fact how to manufacture pure bisantrene dihydrochloride. So that was new innovations there.
Thank you. Okay, while you've got the floor, I might ask you this follow-on question. Do the 2 inactive isomers, and we've had a few questions around this, have 0 effectiveness, or do they have some effectiveness, albeit less than the EE isomer?
Yeah, again, another good question. I showed you on my slides one of the graphs that as you increase the amount of EZ isomer present in solution, you really start to see a drop-off in the activity of the mixture, its ability to kill the cancer cells. So while we can't put a number on it, we can't say it's 0 anti-cancer activity for the EZ isomer, sorry, or the ZZ, because we just can't create pure samples of those compounds. All we can say is that it's absolutely clear that the bisantrene is the most active of the 3 isomers and is possibly the only active one of the 3.
Yeah, and if I can just jump in there as well, it doesn't have to be night and day. It doesn't have to be 0 and 100% between the isomers. All there needs to be is a difference. And clearly it's a very substantial difference. So, as I said, I don't think anyone's going to be able to solve that problem of how to test that definitively. But I think the important thing to take home is that it only really needs to show a difference to be patentable.
Got it. Thank you. Okay.
That answers the utility requirement of a patent. So that shows the use at the end of it.
Right. Okay. Great. Okay, so maybe one for you, Daniel. Did the first trial in Israel, i.e. Nagla 1, use only the EE isomer?
Yes.
Okay. Thank you. Short and sweet. There has been some chat on Hot Copper about transforming the inactive isomers into the active one, EE, just prior to administration. Does that make sense? Or will only the active isomer be delivered to where the drug is administered? Mike?
Yeah, well, as we heard earlier on, having mixtures of isomers is really frowned upon these days by regulatory authorities. So you would have a really hard time getting approval for a drug product that was a mixture. And so there's not really any point in pursuing that angle and trying to develop a drug product that you would do that, convert it all to the EE isomer prior to administration. And in fact, we know that that's not even possible anyway, because if you get a mixture of isomers in solution like that, there's no way you can convert it back to 100% EE. I mean, I mentioned that we did it in the manufacturing, but that's a different story. That was heating it in isopropanol to get it to all convert back to EE. So, I'd say that while it may seem like a good idea, it's just not possible or practical to carry out.
Got it. Okay. Thank you. You would be the expert on these things, so we'll go with your opinion. So, Daniel, how difficult was it for this discovery to be successfully kept confidential for years? And congratulations, it was successfully kept confidential for years.
Um, it's not. I guess if you hire professional people who are all with the single purpose of seeing the success of the company, it's not that difficult. One of the things about Race is that all the employees are shareholders. They're all, they want to see the success of the company. They all believe in it. They work incredibly hard to make it happen. So it's not that surprising that they understand the importance of keeping this secret until it was the right time to patent, which is, the last time. We try and keep it as secret for as long as possible and delay putting in the patent for as long as possible, and that maximizes the lifespan of the patent. So, it's ultimately, yes, it's both easy and difficult. I guess if you hire the right people, it's easy. If you hire the wrong people, it's difficult.
Yeah, and I would add good corporate governance. So well done. Maybe one for you, Pete. Why did you choose to patent all three together rather than having three separate announcements?
Yeah, I know it sort of does look a bit strange to, not try and maximize the number of press releases you can get out. The simple fact of the matter was that each of them contains references to EE bisantrene. And so that would have acted as prior art had we not filed them all at the same time. So they're sort of interrelated. So it might look strange from the outside, but it actually makes perfect sense. It's one of the things that we are always on guard for is what's going into a poster, what's going into a paper, what's going into a presentation, just making sure that we're not talking about something that we're then going to try and patent, and then even a disclosure and investor presentation can act as prior art. It's deemed published, and certainly if it's presented at a scientific conference. So you have to be very, very careful in this space. I mean, everyone in our industry has to do the same thing, but we do monitor very, very closely what data we're letting out and when we're talking about things. And that's one of the reasons why, we knew about this IP, but we couldn't say anything until patents were actually filed, until everything was in. But yeah, that's the reason why we filed the three at the same time.
Thank you. And perhaps while you've got the floor, how confident, because these are patent filings that we're talking about here, how confident can you be that these patents will go to grant?
Well, I think at this stage, you can never say 100% confident because, it's always a bumpy road filing a patent. It's almost like the patent offices are being told to, reduce the number of patents that are getting granted. I think in this case, though, looking at the data and, people online have seen a glimpse of that today. It's pretty strong stuff. And as I said, the fact that 45 years has passed and we're the first people to make this discovery means that it's not exactly blindingly obvious because somebody would have stumbled upon it before. So I think it's pretty robust. I mean, all three of us on here and many others in the company have reviewed the patent filings, and normally, I'm crying my eyes out with boredom after about the first 20 pages. But this one actually was quite compelling. It was. I said to the patent agent, I said it's a ripper. So I think I think we can be very confident. Just a caution: as soon as you get that first examination, there will be all sorts of comments and questions about what we're leadily using. Do we know? There will be questions asked, and it's always an arm wrestle. So just to caution everybody, it's never that looks fine. It'll just be waved through. There'll always be some points that are misunderstood or where we have to make an argument. But that's part of the course. It's what we do all the time. And so we'll wait and see that first examination report. That'll give us a very, very good idea, and obviously we can talk more about that at the time.
Right. Okay, Daniel.
I would just add to that it's not a black and white outcome. There's multiple patents. There's hundreds of claims. We may lose some claim here or there. So it's definitely a grey. Hopefully it'll be close to the black end of grey rather than the white end. But, you just never know on that. So I would expect some claims to get knocked out along the way, but I would imagine all the important claims are very likely, in my opinion, to get up, and certainly the opinion of people far more experienced than me in patent writing share.
Okay, thank you. All right, let's move on. Could it be explained, Daniel, why only a poster presentation was offered at ESMO, given the implied significance of the recent news, which has been known for a long time? My understanding is that an oral presentation was a sign of significance.
Um, the presentation at ESMO is around the mechanism of action of anti-cancer, totally different announcement. So obviously we couldn't put in a poster of this work before the patent goes in, so we can't tell anyone. That would have been a great way of invalidating the patent. So the poster presentation, it's a choice made by the ESMO reviewers. They get far more requests for presentation. ESMO is a clinical conference, so they'll be biased towards clinical trials rather than preclinical results. That's not actually that surprising. So it's just the nature of the way these things go.
Okay. There are a few cute questions in here from shareholders, including this one regarding the blackout, which has been discussed. Is there a real possibility it will be released prior to going to Hong Kong and Berlin? Also, how would you rate the significance of what the blackout is regarding? Thanks for your time, team. Keep up the great work. Daniel?
I've got to love asking a question about a blackout. It means something that we can't talk about because it's price sensitive. So by definition, we can't tell you anything about the reason for the blackout until the blackout is over. So the blackout will end at some point. In my personal opinion, I wish it had ended long ago. Because I've sat there watching the share price go up and not be able to buy. So, and I know Pete shares the same frustration.
Very annoyed.
It's very
annoyed.
It's very annoying to want to buy and not be able to buy.
No, I know, and I've said publicly that I've wanted to buy, and so we can't go into the details of the blackout, but I hope it's over soon. But we're working our way through it.
Okay, we will obviously have to announce any important, material news to ASX. Okay, Are you in discussions, Pete, with a prospective partner presently, looking for a better understanding of what the plans and timeline are here, and please map out the current targeted future milestones?
Right. Oh, crikey, there's quite a bit to unpack there. I might get the future milestones. Throw that to Daniel on the future milestones. But I mean, I've said this before, and I'll just repeat it. Business development is a conversation that stretches out sometimes over many years. As you accumulate data, you go back and you talk to potential partners. And as I said, the more data you have, and the more robust it is, and the better your IP is, the greater the value. So I think we could do a deal tomorrow, but I'm not sure it would be at a valuation that would be we're building enough value for shareholders. Whereas I think we can see certainly that what we're doing, and as the data builds, that we're going to be able to do a much more valuable deal. And and I think this composition of matter patent will expand the horizon of companies who are prepared to talk to us. 20 years exclusivity, and as I said, with maybe the possibility of having 20 years on that phase 3 ready, or even longer with a patent term extension, is pretty rare. So I think we're going to be getting more attention from more companies. I won't add to that, but as I said, we're always talking. We're always talking. We go to conferences, and we're constantly talking. We're sending pieces of information and building that relationship. I mean, I've been in oncology for years, so I've got a database with all of the oncology business development people, so we know who to talk to. I mean, they do tend to move around a bit, so you've got to follow them, But yeah, look, this story is getting more and more exciting, and that just opens up those commercial doors.
Well, I've got another 2 questions for you in a row on partnering. So
Daniel's going to do milestones.
Milestones, go for it. So that was for you, Daniel. I think it was.
Milestones. We couldn't.
Right.
That's a price-sensitive question. We can't answer that outside of an ASX announcement. So, as much as I would love to be able to give you, detailed things on milestones. You'll just have to wait until you see the announcements made.
Though clearly there's a lot going on with the current clinical
What going
study,
on with
right?
current? There's a lot of milestones
Yeah.
already out there. There will be future milestones on other activities as they progress, and we will update our shareholders as soon as we possibly can by the ASX, as is our legal requirement.
Okay, back to you, Pete. So
Yeah.
given your current phase 1 and 2, phase 1/2 trial sites and the new IP position for EE bisantrene, which regions appear most aligned for an initial partnering deal, considering regulatory familiarity, local trial data, and commercial timing?
Yeah, that's a great question. We've talked about exploring regional deals. We think Asia Pacific, clearly with the Korean site and the Hong Kong sites, or Korean sites, that can act as a stepping stone into some of those markets. China is the big one, and we're looking very carefully at that. We're having some discussions with parties who can help with performing transactions. So some groups in Hong Kong. We've announced that we're going up there for the Ignite conference, and we're going to be talking to our principal investigators whilst we're there, but also talking to some other groups, investment groups, and sort of brokers up there. So I think China is one that we haven't quite worked out what our strategy is there yet, but we know there's value there and evolving value. So it's a very, very big user of doxorubicin. We've known that for a long time. So it's an obvious place to go. So a lot of conversations going on around that. But and I think obviously the major markets, they actually might take a little bit longer, getting into Europe and the US. But I think a regional deal somewhere in Asia Pacific, even China, could actually be first.
Okay, follow-on
Without
question.
defining any timelines or anything.
Right, good plan. Okay, follow on though. What would you need in that market to progress from data sharing to a formal partnership?
I think it's building relationships, and again, that takes time. We've identified a number of companies that we want to talk to or are talking to. I've met with a number earlier in the year at ASCO and at AACR. It really is about building that relationship and delivering on milestones, and people seeing that the clinical trials are delivering the safety data, they're delivering the efficacy data, they're delivering cardioprotection data, and just getting that data out there.
Thank you. Okay, this one's for Mike. I would like to ask what protocols are in place for the clinical trials, or perhaps Daniel, depending on how you guys want to play it, to ensure as little light exposure as possible. Are there considerations of the IV line that goes into the patient to reduce light effect? And does body temperature have an impact when EE bisantrene is administered? Who wants to take that?
I'll answer if you want. Yeah, we've done all the tests to make sure that what's delivered to the patient is EE bisantrene, and all the processes are in place to make sure that occurs. Body temperature the temperature at 37 degrees doesn't cause conversion of EE bisantrene. The body, it's the blue light at the spectrum that causes the conversion. And blue light doesn't really penetrate very far into the body, so you just don't get much conversion of EE bisantrene. As long as what goes in is EE bisantrene, it'll stay as EE bisantrene inside the body.
Okay. We've got about 5 minutes left. We've got about 48 questions in the queue. We're not going to get through them all. I'll just say that. But let's go fast and furious. So, Mike, one for you. Well done, team. You've shared a comparison of anti-cancer activity for EE and EE-EZ mixtures. Could you comment on the toxicity profile of EE compared to EE-EZ mixtures, please?
Not really, because we don't have to know about the toxicity of EZ because it's not part of our drug substance or drug product anymore. So we haven't investigated the toxicity of the mixture. My suspicion is that the mixture would be no more toxic possibly, it's not going to be as active in treating cancer because we know EZ is less active. But based on what we know about mechanism of action, we suspect EZ doesn't perform the same way as bisantrene does in terms of the way it kills cancer cells. So we think it's probably a fairly innocuous compound, actually, EZ.
Okay, and then there's a sort of related question. Do the different isomers have different toxicity? We've discussed that, and side effects. So I guess they're useful.
Well, it's possible they do, but it doesn't matter, Jane, because we only put bisantrene in.
Yeah.
It doesn't really matter. It's like could be any other compound. It never goes into the patient, so it doesn't matter. So as long as we deliver bisantrene, we don't have to worry about what properties EZ.
But from a regulatory point of view, clearly the ethos is put as little extraneous compound into a human. It's logical that's going to be the better outcome for the patient. Whether there are any specific side effects of the EZ, we won't know.
Yep. Okay. Quite a few questions, Pete, on how long do we expect the accelerated patent review to take?
It depends on the way you do it, but we're going through that process in Australia, and it takes about 8 weeks. You pay up front for that, but actually that then you get, that's like a down payment for other parts of the process. So I'm not sure we've actually initiated that process yet, but the intention is that we'll do that in the not too distant future.
Okay, I'm going to give this one to Pete. Where do you see the most enduring possibilities for this treatment, and what current future treatment would it be displacing?
Well, it's going to be an add-on, clearly, and where we're focusing at the moment is with doxorubicin, and we're looking at a number of potential indications where that could be advantageous. There are situations like breast cancer, for example, where doxorubicin is used in the neoadjuvant and adjuvant settings, depending on what type of breast cancer the patient has. Clearly, those patients now have very long life expectancy, some of them. I mean, some do succumb quite quickly to the disease. But clearly, that's a group where you've got younger patients living for a long time, where cardiovascular problems are going to be there for a long period of time, affecting the quality of life. So, that will be a setting that we're thinking about, and others. But I think, that's actually an evolving story, and we'll be talking a lot more about that in the future.
Thank you. Okay, we've got 2 minutes to go, so I'm going to offer this last question to Daniel, and then perhaps I might turn back to you, Pete, for some closing comments. Daniel, I missed the window for my bonus options. Any chance of buying those still?
Yeah, yes, you can go to your broker and go online and buy RAC shares, and you will then own your share. So you can spend your money that way. You won't get as many as if you took up your bonus option, unfortunately, but you'll still own a share of RACE. So the answer is no. You cannot. The bonus option expired a year ago. It's more than a year ago.
If I'd been allowed to buy, I would have had bonus options as well. So count yourselves lucky, those who did.
Luckily, we had 82% take up, so I was really pleased. So most people took it up, but obviously that meant that 18% of shareholders didn't take up their options, and I'm sure most of those 18% are kicking themselves now. I hope anyway. So
okay. Let's not rub that in. All right. So there are so many questions left in the queue. I'm sorry that we haven't got through them all, but we will endeavour to get back to people on the primary themes that keep coming through. So keep an eye out for that. With that, Pete, I might hand back to you for any closing remarks and wrap us up.
So again, credit to the team at Race for this really amazing discovery. And again, bear in mind, a lot of people have been working on bisantrene for decades, and they didn't diligently pursue what Mike and the team had done. And I've said before, that Race is it's an amazing group of people. Genuinely, everybody is engaged. Everyone wants to drive forward, get the outcome, deliver things for patients and shareholders. So it's been a lot of fun over the past couple of years, really with this story evolving. I want to thank our shareholders as well. We've got a lot on the line at the moment. We can't do it without your support, so we greatly appreciate that. And hopefully we'll be able to deliver. And just thank everyone online for turning up today and for your interest. Jane, thank you very much indeed. Well compared.
Thank you.
So thank you. Thanks everybody. And as I said, we'll try and get to some of the questions that we weren't able to answer, through Investor Hub or other fora.
Terrific. Okay. Well, with that, I'll add my thanks to all of you for presenting tonight, the many shareholders that were online and investors broadly. And I'll invite you all now to disconnect. Thank you. Have a good night.
Thank you.
Thank you.